everyone, I'm Rosie, Head of Astronomy at Thompson STEM Engagement and also a keen astrobiologist. Today I'm going to give you a brief and hopefully interesting introduction to how we can use bacteria in the search for life beyond Earth. Now, just for background, I'm an astronomer by undergraduate degree and have trained in microbiology whilst completing a PhD at the University of Edinburgh. So hopefully there'll be a good mix of biology and astronomy in this talk. So with that, let's begin. In this talk, I will cover an introduction to astrobiology, a summary of what is life, what are extreme environments, how they can help us assess Martian and icy moon habitability, and finally, how we're searching for life beyond our own solar system via exoplanets. And a good place to begin with this talk is to define the term astrobiology. NASA defines astrobiology as the origin, evolution, distribution and future of life in the universe. It's an extremely multidisciplinary subject, involving astronomers, biologists, chemists, geologists, physicists, engineers and many others, all working together to help further our understanding of the conditions on other planetary bodies and their potential for life. Now, our planet orbits just one star of about 200 billion stars in our galaxy, in a universe of maybe 150 billion galaxies. With a universe so vast, astrobiology also seeks to answer the age-old question: are we alone in the universe? And this question can be exciting at the possibility of discovering new life, but equally terrifying. In the search for life in our weird and wonderful universe, we need to know what to classify as alive. Many organisms beyond our planet, and indeed some found in the extreme environments of our own planet, will seem, for lack of a better term, very alien to us. But from our understanding of life here on Earth, we are able to come up with some key factors that help us define what life is. And defining life is not an easy task, but generally in astrobiology, we tend to go by the Darwinian definition of life, as supported by NASA, which states that life is a self-sustaining chemical system capable of undergoing Darwinian evolution. And Darwinian evolution basically means that all species of organisms arise and develop through natural selection. They do this using small inherited variations that increase the individual's ability to compete, survive and reproduce, as introduced by Charles Darwin. Going by this definition, it's immediately evident that by studying life which is capable of surviving comparable environments here on Earth, it will allow a certain level of predictive power when discussing the possibility of life on other celestial bodies. In addition to the definition of life, we also need to establish what life on Earth has in common. All life on Earth is made up of SHNOPS elements, which are carbon, hydrogen, nitrogen, oxygen, phosphorus and sulfur, with carbon being the backbone of most biological molecules. Life also requires energy to grow, is able to adapt to its environment, can reproduce, can move and can respire. And life on Earth is split broadly into three domains, which can be seen in the image on this slide. This shows the relationship between the domains of life, rooting from the first primordial form, or last universal common ancestor. And the main three domains are bacteria, archaea and eukaryotes, with humans making up here as part of the eukaryotes. Eukaryotes can then be further split into plants, animals, fungi and protists. As we're looking at microorganisms in this talk, we can focus on archaea, bacteria, protists and fungi. So looking closer at some of these microorganisms, the most common that we find on Earth are bacteria, viruses, fungi, algae, protozoa and archaea. Some, like algae, can ruin a fish tank, or fungi, being great on pizza, or others which can send the entire world into quarantine. Now, all of these are microscopic. We need a microscope to be able to see them, with the biggest microorganism being 0.1 to 0.3 millimeters, which is still pretty small. Microorganisms are unicellular, made up of one cell, with the exception of viruses, which are not made out of cells, but single particles called virions. Most commonly in astrobiology, we tend to look at archaea and bacteria, as these are found in some of the harshest environments found on Earth, known as extreme environments, environments too harsh for humans to survive in. So why do we use microbes in the search for life beyond Earth? Well, microorganisms are some of the most hardy and, most importantly, adaptive organisms found on Earth. Due to their simplicity and rapid rate of reproduction, forming millions of cells in a very short time, this allows for large amounts of mutation and adaptation, leading to microorganisms surviving and even thriving in some of the most extreme environments found on planet Earth. These environments are teeming with microbes that can survive the harsh environments. The organisms that inhabit extreme environments tend to fall into two categories: extremophiles or extreme tolerant microorganisms. Extremophiles, from the Greek word philos or love, not only grow under extreme environments but actually thrive within them, and actually require specific extremes such as pH, temperature or salt concentrations to grow. Extreme tolerant organisms are organisms not optimally adapted to survive in extreme environments, but that are still able to grow within them. And archaea is the main group to thrive in extreme environments. Although members of this group are generally less versatile than bacteria and eukaryotes, they're generally quite skilled in adapting to different extreme conditions, frequently holding extremophilic records. Some archaea are among the most extremophilic microorganisms known. For example, the archaeal organism Methanoperos candleri, strain 116, grows at 122 degrees Celsius, the highest recorded temperature, while the genus Picrophilus include the most acidophilic organisms currently known to grow at the ability of pH of 0.06. It's also worth bearing in mind that when we discuss extreme environments, we argue that life in extremes is anthropocentric, that is, from a human perspective. As if microbes could express their views, they would probably call us extreme, as they would not be able to survive in the conditions humans do, for example, your home office environment. Extremophiles were first discovered in these environments in the 1960s within Octopus Springs in Yellowstone National Park, a series of alkaline hot springs. An organism called Thermus aquaticus was found here that could withstand the pH and temperature stresses of the environment. And since then, we've discovered microbes in lots of crazy environments on Earth. For example, sticking to Yellowstone National Park, this is the beautifully coloured Grand Prismatic Springs, named after its rainbow colours. This is the poster boy of astrobiology and extreme environments, with each ring around the lake inhabited by different types of pigmented, temperature-loving microbes. And you can see just how large this environment is from the people on the boardwalk for scale. So this is just one example of an extreme environment, but there are many more out there. For example, the depths of Earth's oceans, or the interior of volcanoes. And many extreme environments actually expose more than one environmental stress at a time. These environments with more than one stress are called multiple extreme environments, and are really, really important in assessing the habitability of an environment. But why are multiple extremes important? Well, let's look at this with relevance to Mars. Very often, studies into extreme environments focus on just one singular stress at a time. For example, low temperatures on Mars, which go as low as minus 150 degrees. However, we don't know how the addition of more than one stress in an environment affects how microbes grow. So mixing those temperature stresses with the high amounts of UV radiation present on Mars due to the lack of a substantial atmosphere, and the toxic salts on the Martian surface, which include perchlorate salts, which are highly oxidizing and even used as rocket fuel. So to really be able to assess the habitability of an environment, on Earth or beyond, we need to be able to understand life's ability to survive in multiple extreme environments. And multiple extreme environments are of course very close to my own heart, as they've been the focus of my PhD studies. I've been looking at how combinations of extreme stresses affect how bacteria grow. And I won't go into too much detail about my PhD, as that's not the focus of this talk. But experiments were carried out in a lab looking at the habitability of aqueous environments on Mars and icy moons. These experiments involve looking at combined stresses of pH, salinity, nutrient limitation, and temperature. By looking at how extremes interact in an environment, we can improve assessments of the habitability of other planetary bodies. So, from all the stresses covered, Mars doesn't appear to be very friendly in terms of a habitat for life, due to its multitude of stresses. But there is hope, as the subsurface of Mars appears to have more favourable conditions for life. This is because the subsurface is shielded from the harsh radiation of the surface, and so may be more favourable. But why do we focus on Mars? Well, Mars has been a major talking point since the 1800s. At that time, Italian astronomer Giovanni Schiaparelli, a director at the Brera Observatory in Milan, began mapping and naming areas of Mars. He observed areas that looked like channels, calling them canali. Now, this was misinterpreted over the years to mean canals, indicating some form of intelligent life on Mars to build such structures. This was further backed by astronomer Percival Lowell, who believed that these lines on Mars were canals used by Martians to carry water from the polar caps down to the equatorial regions. Now, sorry to disappoint, but as Schiaparelli was well aware, this was not the case. And the Mariner 4 spacecraft imaged Mars in 1965, showing no evidence of this intelligent life on Mars. And since then, we've discovered these so-called canals are actually riverbeds on Mars. So we still get an exciting finding from this, as this indicates that there was once stable liquid water on the surface of Mars. And there are two exciting rover missions to Mars in the near future too. We have the joint European Space Agency and Roscosmos ExoMars mission, which has sadly now been pushed back to 2022. But this will have the primary goal of the mission being to determine if there has ever been life on Mars, and to better understand the history of water on the planet. The ExoMars rover, named Rosalind Franklin, includes a drill to access the subsurface of Mars to be able to tell us more about the habitability of that environment. We also have the NASA Mars 2020 mission, which launched last year and is due to land on Mars pretty soon on February the 18th, 2021. The Perseverance rover will seek signs of ancient life and collect rock and soil samples for possible return to Earth. And both of these missions are really exciting, but getting to space is expensive. Is there a way that we could study the habitability of Mars from here on Earth? Well, yes, we can use what are called analogue environments that recreate some of the conditions we believe are present on Mars. One of these environments is the Basque Lakes in British Columbia, a series of small pools filled with high concentrations of magnesium sulfate, Epsom salts, known as brines. Because of the high concentrations of these salts that we find on Mars, this represents an excellent Mars analogue site. And you can see these individual brine pools surrounded by salt crusts. I was lucky enough to visit this environment during my PhD for fieldwork, and of course we took a plushy Mars with us to the Mars analogue site. But now, back towards the title of this talk. How can bacteria on Earth help us in the search for life on other planets or even moons? Well, one way is by using these analogue environments to find bacteria that can survive the conditions we know of on Mars or icy moons. We can then try to recreate some of these conditions in a lab to see if the conditions affect how and if the bacteria grow. And we have a multitude of these analogue sites here on Earth, which you can see in this figure, which match to different parts of Mars so we can explore how its geology may affect its habitability. These environments include field sites in Iceland, which are highly volcanic regions and can be used as an analogue to the volcanic Tharsis regions on Mars. The Danakil Depression in Ethiopia, which is geothermally heated to be one of Earth's hottest environments during the summer, and could be an analogue to past conditions on Mars. There's the Basque Lakes, which I previously mentioned. There's also Kangerlussuaq, which is an area of permafrost in Greenland, which can be used as an analogue to the poles of Mars. the Makati salt pans, which were once a really salty lake where the water evaporated and left only salt deposits behind, and the Rio Tinto River, which is blood red in colour and has similar salts and minerals to those detected on the Martian surface. These are just a few, but all of these are very exciting environments and can hopefully tell us more about the habitability of past and present Mars. So, following this, what would any potential life on Mars need to be tolerant to? Well, firstly, it would have to survive the high levels of radiation, also the low temperatures, an alkaline soil with pH indicated by the Curiosity rover in 2008, and high concentrations of many different salts. But on from Mars, as there are a few other places to look at in our search for life in the solar system, and on to the outer solar system, because we think this is where there are several places that are promising in the search for life beyond Earth. These include the icy moons of the gas giants. There's the moon of Saturn, Enceladus, and the moon of Jupiter, Europa, which are both icy moons thought to have liquid water oceans beneath their surface. Few worlds in our solar system are as compelling as Saturn's icy moon, Enceladus. A handful of worlds are thought to have liquid water oceans beneath their frozen shell, but Enceladus sprays its ocean out into space, where spacecraft can reach out and sample it. From these samples, we've determined that Enceladus has most of the chemical ingredients needed for life, and likely has hydrothermal vents, spewing out hot mineral-rich water out of its ocean. With its global ocean, unique chemistry and internal heat, Enceladus has become a promising lead in our search for life beyond Earth. And in the search for life beyond Earth, we can't forget Jupiter's moon, Europa. Decades ago, science fiction offered a hypothetical scenario. What if alien life were thriving in an ocean beneath the icy surface of Jupiter's moon, Europa? This notion pulled Europa out of obscurity and into the limelight where it's remained, stoking the imaginations of people both within and outside of the science community, who all fantasise about humans discovering life beyond Earth. And that fantasy may actually be grounded in reality. Ground-based telescopes on Earth, along with the Galileo spacecraft and space telescopes, have increased our confidence for a subsurface liquid water ocean on Europa. We think that Europa's ice shell is 10 to 15 miles thick, floating on an ocean 40 to 100 miles deep. So while Europa is only 1/4 the diameter of the Earth, its ocean may actually contain twice as much water as all of Earth's oceans combined. This does raise a bit of a problem, however, as if we observed life here on Earth, it was long believed that the life cycle required sunlight in order to be able to be sustained, a source of energy. With the depth of the oceans and the thickness of ice covering Europa, there could be no chance of the development of life, right? Well, in 1977, while exploring an oceanic spreading ridge near the Galapagos Islands, scientists discovered hydrothermal vents. To their amazement, they also found that the hydrothermal vents were surrounded by large numbers of organisms that had never been seen before. These organisms were drawing their energy from the hydrogen sulfide gas pouring out of these vents. Following this, if a similar mechanism exists at the bottom of Europa's ocean, then life could be a real possibility, surviving using similar mechanisms. We can summarise the main stresses encountered on the icy moons would be low temperatures, high levels of salt, capable of keeping their oceans liquid at such low temperatures, and pressure caused by the thick ice layer above. As these moons are so far away, there are actually analogue environments here on Earth, just like Mars, that we can use to study them. One of these is Lake Vostok, a subglacial lake in Antarctica which represents a good analogue to the icy moons. Temperatures in this lake are approximately -2 degrees, but the lake remains liquid due to a 2-mile thick ice sheet above it, exerting high amounts of pressure and keeping the water liquid. If any life survives in this environment, it would have to depend on a different energy source from sunlight. And it's believed that organisms may get their energy from chemicals in rocks in the environment, or there may be hydrothermal systems present, just like those detected in the Galapagos Islands. And the best way to determine if there is life on Enceladus or Europa is to actually go there. There are currently no planned missions to Enceladus, but there are several upcoming missions to investigate Europa. These include the European Space Agency JUICE mission, due to launch in 2022, which will fly by Europa alongside the other moons of Jupiter, Ganymede and Callisto. Then after this, the NASA Europa Clipper is due to launch in 2024 to investigate and confirm surface conditions on Europa, hopefully followed by the NASA Europa Lander, which is still not a confirmed project but a lot of work has gone into. And the Europa Lander would allow us to access Europa's ocean to analyse the conditions and understand its potential for life. But there is another moon that we want to consider in the outer solar system that does have a potential for life. And this is Saturn's largest moon, Titan, and is an extraordinary and exceptional world. Among our solar system's more than 150 known moons, Titan is the only one with a substantial atmosphere. And of all of the places in the solar system, Titan is the only place beside Earth known to have liquids in the forms of rivers, lakes and seas on its surface. And although this might sound promising for life at first, these rivers, lakes and seas are actually made up of liquid hydrocarbons, like methane and ethane instead of water. We do, however, think that Titan may have a subsurface water ocean, like Europa and Enceladus. So perhaps Titan's subsurface water could be a place to harbour life as we know it, while its surface, lakes and seas of liquid hydrocarbons could conceivably harbour life that uses different chemistry than we're used to. That is, life as we don't know it, yet. Titan could also be a lifeless world, but that's a less exciting option. Now there's a large amount of methane on Titan, and sadly, these creatures, cows, aren't the culprits. We think that maybe life similar to methanogens on Earth, which are archaea that produce methane as part of anaerobic respiration, could plausibly survive on Titan. Although all living things on Earth, including methanogens, use liquid water as a solvent, it's conceivable that life on Titan might instead use liquid hydrocarbons such as methane or ethane. The fact that Titan appears to have lakes of liquid ethane or methane on its surface, as well as these rivers and seas, suggests that it could support hypothetical non-water-based life. It's been speculated that life could exist in the liquid methane and ethane that forms these rivers and lakes on Titan's surface, just as organisms on Earth live in water. Such hypothetical creatures would take in hydrogen in place of oxygen, react it with acetylene instead of glucose, and produce methane instead of carbon dioxide. By comparison, some methanogens on Earth obtain energy by reacting hydrogen with carbon dioxide, producing methane and water. And to find out more about the habitability of Titan, the Dragonfly mission will sample and examine sites around Saturn's icy moon. The mission, due to launch in 2026, will arrive in 2034. The rotorcraft, designed to look like a dragonfly, will fly to dozens of promising locations on Titan, looking for prebiotic chemical processes common on both Titan and Earth. And Dragonfly marks the first time NASA will fly a multi-rotor vehicle for science on another planet. It has 8 rotors and it flies like a large drone. And Titan can actually act as an analogue to the very early Earth, and can provide clues as to how life may have arisen on our own planet. During its 2.7-year baseline mission, Dragonfly will explore diverse environments, from organic dunes to the floor of an impact crater, where liquid water and complex organic materials key to life once existed together for possibly tens of thousands of years. Its instruments will study how far prebiotic chemistry may have progressed, and will also investigate the moon's atmospheric and surface properties, and its subsurface ocean and liquid reservoirs. Additionally and importantly, the instruments will also search for chemical evidence of past or extant life. And finally now, to zoom out beyond our solar system and take a look at exoplanets, planets that orbit stars other than our own Sun. The James Webb Space Telescope, due to launch in October of 2021, will be able to tell us more about potentially habitable worlds outside of our solar system. And the James Webb Space Telescope will tell us more about the atmospheres of extrasolar planets, and perhaps even find the building blocks of life elsewhere in the universe. And it will do this using a technique called spectroscopy, which is simply the science of measuring the intensity of light at different wavelengths. The graphical representations of these measurements are called spectra, and they're the key to unlocking the composition of exoplanet atmospheres. When a planet passes in front of a star, the starlight passes through the planet's atmosphere. If, for example, the planet has sodium in its atmosphere, the spectrum of the star, added to that of the planet, will have what we call an absorption line in place of the spectra, where it would expect to see sodium. This is because the elements and molecules absorb light at characteristic energies, and this is how we know where in a spectrum we might expect to see the signature of sodium, if it is present. The James Webb Space Telescope will also analyse locations a bit closer to home, and will investigate the plumes of Enceladus and Europa. The ultimate goal of the Webb Space Telescope is to find a planet with an atmosphere similar to that of Earth, and detect elements indicative to life, such as the Schnops elements that I mentioned earlier. And with that, I hope you enjoyed a quick guide to astrobiology and how we can use bacteria in the search for life beyond Earth. Thank you for listening, and make sure to check out next week's set of videos where Tony Thompson will start a set of chemistry demonstrations. But with that, please share, like and subscribe, and carry on learning. Bye!